My name is Lalita Ramakrishnan. I'm a professor of immunology and infectious diseases at the University of Cambridge. And in this lecture, I'm going to continue to tell you a little bit more about tuberculosis, focusing on a structure called the tubercle. Now, just to recapitulate the lifestyle or the life cycle of Mycobacterium tuberculosis, which is the causative agent of tuberculosis, the bacterium that causes it. The bacteria are exhaled in coughs by people who are infected, get inhaled by individuals near them, and then they enter cells that are called macrophages. But what happens next is that they induce these macrophages to form structures called granulomas. And these granulomas can become quite elaborate. At first, they're comprised just of macrophages, but then many other immune cells come in and they can form a fairly complex structure. And another thing to note is that the macrophages within this granuloma undergo a specialized differentiation called epithelioid transformation, where they form these finger-like interdigitated projections between each other to form a very compact organized structure. Now, this structure, pathologists call these structures granulomas. And granulomas are associated with many diseases, both infectious and non-infectious, and they can often be quite pathological. But the single biggest cause of granulomas is tuberculosis. And in fact, granulomas were first discovered in the context of tuberculosis in 1679, some 200 years before the bacterium, Mycobacterium tuberculosis, was discovered. And in fact, the structure used to be called a tubercle, and obviously both the bacterium and the disease are named for the granuloma. But turns out that granulomas are very primitive structures. They probably evolved. They're present even in lower vertebrates in a more primitive form, and they probably evolved to wall off foreign bodies. You could imagine a thorn being circumscribed by such a structure until it's sort of dissolved. But these so-called foreign body granulomas have a very low turnover of macrophages. The macrophages just come and sit there, and it's not a particularly inflammatory structure, or so it's thought. In contrast, the types of granulomas that form with tuberculosis and pretty much any medically significant granuloma tend to be high turnover granulomas where there's a rapid death of macrophages and a repopulation by new ones, and they can be very inflammatory structures. There are many traditional animal models that are used to study TB. The oldest ones are the rabbit and the guinea pig, which were used by Villemin and Koch, respectively, at the time that they discovered TB. They used these animals to pass the bacterium from one animal to another to show that it was associated with TB. The most commonly used model now is the mouse, and this makes a lot of sense because mice have a wonderful array of immunological and genetic tools. One issue is that most mouse strains don't develop the nice tight granulomas that are associated with human disease, but there are some recently identified mouse strains that do. And so those could actually be quite good. Another more recently used model is the non-human primate. And this is a good model because it really recapitulates human disease. But the problem is, of course, they're expensive, there are ethical considerations, and obviously cannot be used widely. So on this backdrop, my story and my engagement with TB and the granuloma came when, as a postdoctoral fellow at UCSF, I was a clinical infectious disease fellow and had to do a postdoctoral fellowship, I approached Stanley Falco at Stanford to go to his lab and study TB, which he didn't study at the time, but he studied many other bacterial pathogens. And Stanley said to me, forget it. I don't have the specialized containment facilities you need to study TB, which is a human aerosol pathogen. And besides, he said, TB grows so slowly, I'll be dead before you get your first result. And that was in 1991, and I'm happy to say he's still alive and quite well. So what he told me, he gave me an insight, and he said, look, there are other strains of Mycobacteria, there are other species of Mycobacteria that are pathogenic in other animals, that are natural pathogens of other animals. And he said, I'm pretty sure there are these ones of marine life, of fish, because I've seen people get them who were fishermen. He had worked at Brown and knew that Portuguese fishermen got this disease, mycobacterial disease, on their digits and on their soft tissues. And so I went off to the UCSF library and looked at this very classic manual. It's called Bergey's Manual of Systemic Bacteriology. And I found what he was talking about. He was probably talking about a bacterium called Mycobacterium marinum that was thought to be a close relative of human TB, of the human TB bacterium, and it gave fish TB. It turns out that it also infects humans, and this has been known since the 50s, and I can personally attest to it. And it gives humans disease on their extremities, as Stanley already knew, and many clinicians, particularly dermatologists and infectious disease clinicians, already know this. But if you look inside that lesion, you'll see a classic granuloma, and actually in many cases it can be indistinguishable from the granulomas caused by the human TB bacterium. But of course, we know that this bacterium also infects fish, and it was first identified to do so in the Philadelphia Aquarium, where in 1926, fish were dying of some mysterious wasting disease, very similar to human TB. And when they tried to culture these fish to see what bacterium they had, why were they dying, they couldn't culture anything. But when they looked at the fish by histology, they could see these classic red snapper bacteria that looked very much like TB. And then Aronson had the bright idea to try to do the cultures at a low temperature that was commensurate with the low body temperature of the fish. And then he was able to culture Mycobacterium marinum. And since then, we've had Mycobacterium marinum sequenced at the Sanger Center, and it turns out to be the closest genetic relative of the human TB bacterium, so I guess we also got quite lucky. And it turns out that Mycobacterium marinum also infects zebrafish. Zebrafish are a pet organism of developmental biologists and are a natural host to Mycobacterium marinum. So I've got for you here down in the bottom panel a human TB granuloma stained by hematoxylin and eosin, which will only stain the host cells but not the bacteria. And what you can see is that you've got a nice organized structure, which is cellular, as evidenced by blue nuclei on the edges, but in the center where that arrowhead is, you'll see that the structures become acellular because it's undergone necrosis, just as we know that human TB granulomas do. But let's take a look now at a zebrafish granuloma. And in this granuloma, you can see, which I've stained here with a stain that also stains the bacteria. You can see that it looks very similar, and it's a nice cellular organized structure. And you can see that there are a few bacteria within macrophages, but where the macrophages have necrosed, there are tons of bacteria, which is exactly what you would expect. But the great feature of the zebrafish that makes them so enticing to developmental biologists is that they have a prolonged larval phase when they're transparent. And so you can actually watch things happen, and people watch developmental processes happen. But so we asked, well, can we put in bacteria and watch infection happen? And so they have a cavity that's called the hindbrain ventricle, which is the equivalent of something in our brain, close to our brain, called the fourth ventricle. And so we put in some bacteria there. I've shown it to you with an arrowhead there. And what we saw very quickly was that macrophages came, and you'll see the macrophages sort of chasing after the bacterium like a cat after a mouse, and eventually you'll see this macrophage gets it. And there you've got an infected macrophage. You can now follow these infected macrophages out of the cavity. This is a few days later, and you can see that it's just moseying along. The bacteria have grown in the macrophage, and because it's a permissive macrophage for the bacterium, and there it is. But what was really exciting to us was that you could see within a few days a granuloma form. And here you can see that we've got a granuloma that's already formed. And what you're going to see where that white arrow is, a new uninfected macrophage is going to come, and then it's going to enter the structure. So watch this. See, there it comes. And it's going to squeeze its way in between and get in there. So the granuloma is a highly chemotactic structure that is recruiting new macrophages to come to it. And then we could show all this by engineering fish that were transgenic, so that they had green fluorescent macrophages and red fluorescent neutrophils, which is another cell type that is somewhat involved in granulomas, but not as much as macrophages. And now we've infected the fish with blue fluorescent bacteria, and you can see that we've got a nice tight bona fide epithelioid granuloma with infected macrophages. So this was good. As we were developing this model, my colleague and friend David Sherman made a suggestion to us. People have been searching for virulence determinants in Mycobacterium, and one exciting discovery was that a specialized secretion system called the ESX-1 or RD1 locus, which I've shown in white in that top panel, the white genes in the top panel, were involved in virulence. And this was very exciting because it turns out that this was the locus that was missing in the attenuated vaccine strain BCG that was made by serial passage in the 1920s, and now we finally knew the molecular basis of its attenuation. So Mycobacterium marinum, not surprisingly, has a locus that looks virtually identical. And David kept telling me, look, make a mutation in this, and let's see how it really works, because everyone knows it's attenuated, but a lot of these animal models are black boxes because you only get to see the end result. And he could see that we would be able to get some insights about the actual sequence of what was different. And when I was a bit slow to do this, he actually had someone in his lab make the mutant for me, and he gave it to us and said, take this. So at this point, we were sort of shamed into doing this quickly, and we was Hannah Volkman, who had joined my lab as a graduate student. And Hannah showed very quickly that, yes, if you put this mutant into zebrafish larvae, it was attenuated. The animals didn't die, and if you looked at the bacterial counts, you could see that the bacteria didn't grow as well. So this was good because it showed us that it was behaving just like you would expect. But here came the surprise. And by this point, Hannah had recruited some of her colleagues, Dana Beery on the left, who was a technician in the lab, and Hilary Clay, a graduate student who was Hannah's very good friend. And she got them to join in this quest to see what was going on. And what they found was something quite interesting, because if you look at the fish on top, they are infected with wild-type bacterium. And if you look at the close-up on the top right, you can see that a nice big granuloma has formed. But if you look at the mutant, what you can see is that even if you inject many, many more bacteria just to compensate so that you get as many bacteria as with the wild type, the macrophages pack up with the bacteria, as you see on that bottom right panel, but they don't form granulomas. Now, this seemed opposite of what you'd expect, because if granulomas are good for the host, as what everyone in TB, in the field of TB thought, people have thought that the granuloma is a critical host protective structure that walls off the bacteria, and while it's not always successful in eradicating the bacteria, it sure as heck tries to do so and is pretty good at it. Now, if that's the case, then we should see more granuloma formation with that mutant, but we saw less. So Hannah took a close look at this, and she was able to observe fish as the granuloma formed by serial imaging. And what she showed was that when the granuloma formed, the number of infected macrophages went up dramatically, as did the number of bacteria. So the granuloma was actually promoting growth rather than restricting growth. So why might this be? Because here we are saying that this immunological structure that really should be killing the bacteria is actually promoting growth. And the answer to this came both from Hannah's work and from a new graduate student who joined, an MD PhD student, Muse Davis. And what we found was happening was that when there's an infected macrophage, when new macrophages come to it, for some reason, if they have that ESX-1 locus, the bacteria are spreading quickly from macrophage to macrophage. You can see within 48 hours, you've gone from one infected macrophage in that top left panel to many infected macrophages. Whereas if you didn't have that locus, if the bacterium didn't have that locus, then that one macrophage just remains one great big macrophage, and the bacteria are just growing in it. But obviously, they're not doing as well as if they can spread to new macrophages. And so it turned out that the bacterium is using this locus to spread from one macrophage to another. Now, how does this happen? What Muse found was that if the initial macrophage was infected with bacteria that contained this locus, then somehow that macrophage was able to exert a rapidly chemotactic effect on macrophages around it, or even far away, so that they came. And you can see that the macrophages in the top panel have these protrusions that reflect that they're highly chemotactic and are responding to a chemotactic gradient and are racing into this structure. In contrast, if that initial macrophage didn't have this locus, then the incoming macrophages are coming in very, very slowly, and they clearly are not experiencing a chemotactic gradient. They don't have that great big protrusion. And even once they get into the granuloma, they behave very differently. The wild-type macrophages move far and wide within the granuloma and rapidly, whereas the mutant macrophages, the few that do come, are just sort of sitting there like bumps on a log. And this is the kind of movie that gave us that insight that I just talked to you about, that macrophages are exploited by the bacteria to spread from cell to cell. So what happens is that when a given infected macrophage packs up with bacteria, because the bacteria can grow within it, it undergoes an apoptotic death where it's dead, but it's preserved its membranes. And now what happens is this dead cell is recognized by the incoming macrophages that come and eat it. And I'm going to show you an example here of where one dead macrophage with the white arrow is going to be engulfed by an incoming macrophage. So watch this happen. you're going to watch it, it's going to come from below. It's kind of like that movie Jaws where the shark comes from below, and look at it. It's eating it bite by bite. And this is why macrophages might be called what they are, macrophage for big eater. And now you've got this macrophage has been eaten by a new macrophage. But you're going to say, wait a minute, why would this spread the infection? You've gone from one macrophage to another macrophage. So all you've done is conserve the bacteria. But it turns out, when Muse looked closely, that on average, a given macrophage, given infected dead macrophage, was eaten on average by 2.3 macrophages every 24 hours. And so you can imagine, you can see how the bug is using the macrophage to expand its numbers. And not only that, but we showed that the bacterium also induces the death of the macrophages. Here's a tunnel stain on the left, and this death, there are probably many bacterial determinants that do this, but one of them is that self-same locus, ESX-1, that also induces the macrophages to come. So it's got a two-pronged effect. It's inducing death of the infected macrophage, and separately it's recruiting new macrophages to come to it so that they can engulf the dead macrophage and produce infection. So to summarize this, let's take a look at what happens in the mutant first. Because in the mutant, the granuloma might actually be functioning as a host protective structure as it might be meant to be. You've got an infected macrophage. It dies at a slow rate. New macrophages are slowly recruited at a respectable pace. And now they can eat the new macrophage one by one on one, and there's some time for the cells to also kill the dying cell to also kill the bacteria before they're eaten. So you could imagine there's an attrition of bacterial infection. This might be why the BCG vaccine strain is attenuated. But paradoxically, instead of sort of thwarting these host protective processes, as you might imagine a pathogenic bacterium might do, the pathogen actually accelerates these processes. So it converts them from being a host protective to a host detrimental process. So simply by speeding up the rate of cell death and speeding up the recruitment of new macrophages, it's just using the macrophage niche to spread from cell to cell and therefore expand itself in the granuloma. And this is quite a nifty thing to do, I think. So, okay, but the question now is how does this ESX-1 locus induce the recruitment of new macrophages? And so for this part, Hannah was joined by Tamara Pozos, who was a pediatric infectious diseases fellow who joined the lab. And together, they did a microarray where they looked at fish that were infected with wild type or mutant bacteria to identify host genes that might be different between the two. And the gene that stuck out was matrix metalloproteinase 9. And this is a extracellular, an enzyme of, that remodels the extracellular matrix. And they were even able to show, not only by transcriptional analyses, but also by doing a gelatinase assay on the fish for the actual activity of this enzyme, that MMP9 was induced upon wild-type infection, but not upon mutant infection. Now, that's fine, but if induction of MMP9 is responsible for the ESX-mediated acceleration of macrophage recruitment, then if you make an MMP9 mutant, that mutant should be attenuated even with wild-type infection. And sure enough, when they looked, they saw that the MMP9 mutant, which is shown on the bottom there, that fish, was attenuated for infection, and it had very few granulomas. So it was behaving just like the bacterial mutant, and therefore it was the partner. So this was nice, but then we started to look into what MMP9 does, and then it turns out that MMP9 is involved in the pathogenesis of arthritis and cancers, and other inflammatory conditions. And often in those cases, it's a macrophage that is the bad actor that is making MMP9 and causing trouble in these lesions. So of course we thought, well, okay, a macrophage gets infected with the bacteria, it now induces MMP9 in the macrophage, and now that is secreted and calls in new macrophages. But when we did in situ analyses to look at where the MMP9 was being made, here is a granuloma, and the MMP9 is labeled green and the macrophages are labeled red. And what you can see is that the MMP9 is not in the macrophages of the granuloma, but rather it's in the epithelial cells surrounding the granuloma. And so what seems to be happening is that an infected macrophage secretes something from that secretion system that goes and talks to the epithelial cell and induces it to make MMP9. And the MMP9 now calls in new macrophages. And this strategy was called subversion from the sidelines by my friend and colleague Bill Bishai at Hopkins, and I rather like how he put it. So why might that be? You could imagine that the bacterium wants to tamp down immunity in the macrophage itself because it has to survive in there. So it's doing that, and meanwhile it's inducing an inflammatory program in a neighboring cell so that it can bring more macrophages, infect, and then subvert them. Okay. So this is how the bacterium uses the innate immune phase of the granuloma to promote its growth and expansion. Of course, then the granuloma matures and other things happen. And as I told you, one of the things that happens is epithelioid transformation, these tight interdigitated projections. That too has been known for 100 years or so, and that very reasonably was thought to be a host protective mechanism that would sort of wall off the bacteria and perhaps be somehow help the host despite all these strategies of the bacterium. Well, very recently, work from David Tobin's lab, also done in the zebrafish, that this too turns out not to be the case. Mark Cronin and David Tobin have shown that epithelioid transformation of the macrophage is also something that the bacterium is benefiting from. If they inhibit it, then they get less infection than if it's there. And of course, they're working out the details of how this might be, but what it's telling you is that practically every step that we might predict would help the host can be taken advantage of by the bacterium. So in my first lecture, I told you that most people actually clear infection, and they do so after the adaptive phase of the granuloma has kicked in. So it's very clear that at some point the granuloma can fight back and can eradicate or at least suppress infection. And many people who specialize in the areas of adaptive immunity and TB are working on this. But I want to close by saying that while the adaptive immunologists may not know as much about this as they want to, and they're working hard to figure it out, the bacterium seems to know quite a bit about this, these immune mechanisms, because what these people who worked on it so far can tell you is that it tries really hard to delay and inhibit adaptive immunity so that the adaptive immune elements that come into the granuloma do so late. And this gives time for the mechanisms that I've just been telling you about to help bacteria expand in the innate context. I'll close by thanking the many people whose research I've described to you. They're both students and from my lab, as well as colleagues and collaborators from outside my lab and indeed from across the world. Thank you.